Hi, and welcome to a quick tutorial on Mendel's crosses. Gregor Mendel was an Augustinian monk back in the 1800s, and he's the first Western scientist who figured out how genetics works. So I'm going to step you through a little bit of his thought processes in this video, and we're going to learn about the 3 generations of crosses that he used to figure out how genetics works. Mendel used pea plants, and pea plants are great to use because they are quick to reproduce. You can get several generations in a single growing season, and they have very distinct traits. The trait that we're going to use is flower color. In pea plants, flowers can either be purple or white. And right now, we know that purple is dominant to white, but I'm gonna show you why that exists in a second. So let's just use a capital P for purple and a lowercase p for white. I'm exaggerating the difference between the capital and the lowercase because it's kind of hard to tell the difference if you don't kind of exaggerate the difference between a capital P and a lowercase p. So the first series of individuals in a cross he called the parental generation. And this is the parental generation, not the parent generation. You'll find out a little bit more about this as we go on, but basically anything that reproduces can be a parent. But the parental generation has a very specific definition. And that definition is that they're between 2 true breeders. Now, another word for true breeder is homozygous, of opposite phenotype. So in the cross of the flowers, he would have made sure that he had a purple plant that was a true breeder, and he would have crossed that with a white plant that was a true breeder. If you might have seen some of my other videos where I talk about how to do Mendelian genetics, and if you do that, you'll notice that I am now doing the table of gametes. I just like to show how meiosis happens when we're looking at our genetic crosses. So I'm showing you the gametes that come out of an individual. This one's making the male gametes. We'll just assign this one to the female gametes. And that's the symbol for female. And we're just gonna imagine those are eggs. So sperm and eggs, right? Remember that sperm fertilizes eggs. So if we take these 2 individuals and we make these gametes, we can then put them into a Punnett square. And the Punnett square is the prediction of what should happen if you have these 2 individuals mating. And of course, we only have the allele for the dominant trait in that one, and this female here has only the allele for the recessive trait. So when we say when sperm meets egg, we have this outcome. And notice that all of them are heterozygous. But if you look at the phenotype of that cross between the purple and white parentals, it turned out that they were 100% of the offspring were purple. And this is how we know, sorry, that was off screen for a second. This is how we know that purple is dominant, because whatever shows up in this generation, the offspring of the parental generation, that's the dominant phenotype. It just shows up. Every single one of them has it. So back here, this is why I knew that purple was dominant and white was recessive, only because that's what showed up in the F1. And this generation here, these ones that we predicted that are the offspring of the parentals, are known as the F1 generation. So let's take a look at what would happen if 2 F1s were crossed together. And that's actually what Mendel did. This is going to be the 2nd generation in a series of crosses where we look at what happens when you cross 2 of those F1 generations. So F1 generation, if you'll remember from the last piece of paper that I showed, both of those individuals were heterozygous. So we're just crossing those 2 heterozygotes together. They would have been siblings in Mendel's crosses. He would have taken 2 siblings. And we're going to do that table of gametes again just to show what meiosis looks like. So that's a capital P, and here's a lowercase p. And the same thing for the eggs, a capital P and a lowercase p. So when we put this into a Punnett square, and remember that Punnett squares are our predictions about what should happen in the next generation. We put the sperm over here and the eggs over here, and we just fill in what would happen if this sperm met with this egg, what would happen if this sperm met with this egg. And that would be homozygous dominant. If this sperm met with this egg, it would look like that. If this sperm met with this egg right here, it would look like that. Again, we put the capital P first, and then if this sperm met with that egg, it would look like that. So in this generation, we end up getting a 3 purple to 1 white flower, because as long as you have one dominant letter, you show the dominant phenotype. So 1, 2, 3 dominants, and 1 recessive. Again, this is just a prediction. When Mendel actually did his crosses, he got something in the neighborhood of 3 to 1, and he just kind of averaged it out. This generation right in here, these 4 here, are known as the F2 generation. In the F2 generation, if you're following all 3 generations in Mendel's crosses, the parental generation, which had a very specific definition of 2 true breeders of opposite phenotype, the F1 generation, which every single one of them shows the dominant phenotype, and then the F2 generation, where you get a 3 to 1 ratio. The thing that was fascinating to Mendel was that the white ones showed back up again. And that's when he figured out that, oh, there's something about the white ones that's hidden if ever there's a purple allele around. So he called this recessive. This is the recessive phenotype. You've got the 2 recessive alleles. This is the dominant phenotype. And you either have, as we've learned here, 2 dominant alleles, or you're heterozygous. But again, each of the generations was real specific. So the F1 generation, they're always heterozygous. In the F2 generation, you've got this interesting ratio. Hope this helps you understand Mendelian genetics and how Mendel came up with his ideas of inheritance.